DNA replication. So how do we form new DNA? So for us, DNA is semi-conservative. So that means one of the parental strands is used as a template for the daughter strand. So each new double strand of DNA that we synthesize, one of the strands was from the parent cell. So there is one parent cell in each of the two resulting double-stranded DNA, while the other strand is newly synthesized DNA. So that's why it's called semi-conservative. So it's not all parent DNA and it's not all brand new DNA. It's half parent DNA and then half newly synthesized DNA. And then, so how does DNA replication begin? So the first thing that happens is helicase, the enzyme unwinds the DNA to make two single-stranded DNA strands, and they started at origins of replication. So DNA is double-stranded, and helicase works to unwind the DNA to create two single strands, so the other proteins and enzymes can get to the DNA to replicate it. And then, so the next thing that happens is single-stranded DNA binding proteins. They bind to the unraveled strand after the DNA helicase unwinds them, and this is to prevent reassociation of DNA and degradation by nuclease. So these single-stranded DNA binding proteins, they bind to the two single strands in order to stop them from associating back. So they don't want it to reform a double-stranded DNA. They want to keep it at two single strands. And then the next thing is DNA topoisomerase. So they work to create negative supercoils to decrease stress from supercoiling. So when the helicase unwinds the DNA, it strains the DNA when it unwinds it because it creates stress at the far ends of the DNA. So that's called supercoiling, and DNA topoisomerase works to relieve that stress by nicking either one or two strands at specific spots on the DNA, and this works to prevent supercoiling. It relieves the stress. So instead of one really long strand that's being pushed to the edges when it unravels, it has little nicks in it that are then resealed to prevent supercoiling. And so when DNA helicase unwinds the DNA, it does it at origins of replication, and then this creates replication forks. So you can see on the picture on the right, the fork works in one direction, and that is the direction of the leading strand. So in eukaryotic chromosomes, there are multiple origins of replication forks, because our DNA, eukaryotic DNA, is a lot larger than bacterial or prokaryotic chromosomes. So multiple origins creates multiple leading strands and Okazaki fragments. This is to speed up the process of DNA replication, while bacterial DNA, it's just a single loop of DNA, and there's not much DNA in it, so they only need one single origin, while eukaryotic DNA, which is a lot larger, they require multiple origins and multiple replication forks in order to speed up the process. And like I said before, whenever there's a replication fork, there's a leading strand where the DNA is replicated in the direction of the replication fork. So it's just a one continuous strand of replication, while the Okazaki Okazaki fragments are created in the lagging strand. So since the lagging strand is facing away from the origin of replication, each time the replication fork moves in the 3' to the 5' direction, or there's multiple Okazaki fragments needed because of the replication fork keeps moving in one direction. So the Okazaki fragments are created each time the DNA is made. Yeah. So DNA polymerase is the one responsible for creating new DNA. So it reads the parental strand from the 3' to 5' position, while it makes the new strand in the 5' to 3' position. So since the leading strand is always being read in the 3' to 5' position, it just keeps making one long continuous strand. But the lagging strand, since it's anti-parallel, it has to be read multiple times by DNA polymerase, and that's why there's all these mini Okazaki fragments. Okay. But each Okazaki fragment needs a primase to make an RNA primer. So while the leading strand only needs one RNA primer, since it's one long continuous strand, the Okazaki fragments need multiple RNA primers because of how it's made up of many small strands of DNA. So each time a DNA is added, a DNA polymerase adds a 5' deoxyribonucleotide triphosphate. So each new nucleotide added is either a DATP, DCTP, DGTP, or DTTP, which represents the 4 nitrogenous bases. So there's 4 different nucleotides added. And each time the nucleotide is added, a new phosphodiester bond is made between the nucleotide that was already added and the new nucleotide that's being added, and a pyrophosphate is released. The pyrophosphate is released because DNA replication is it requires energy, and like I said before, the negative repulsion of the phosphates on the nucleotides, that's the energy needed to form the DNA. So the pyrophosphate is released in order to generate the energy needed to form that phosphodiester bond. And another function of DNA polymerase is it removes the RNA primer that was added by primase. So once it finishes replicating the DNA, it'll remove the RNA primer and replace it with DNA nucleotides instead. So for the lagging strand, it only does this one. For the leading strand, it only does this once, while the lagging strand, it has to do it multiple times. And then on the right is just I like this picture of showing all the things that's happening during DNA replication. Then finally, once DNA polymerase is finished replicating all the DNA ligase seals the ends of the DNA together so it doesn't unravel. So it keeps the double-strand DNA together. But like I said before, DNA polymerase, it can't just add on to, it can't just start replicating DNA. It needs a primer. And since at the end, at the ends of the chromosomes, the strands of DNA, you can't add a primer to the very ends because there's not enough space for there to be a primer and then to add DNA. So at the ends of the chromosomes, there are things called telomeres. So these are still DNA. They're repeating DNA sequences at the end that don't really code for any genes. They're just there to protect the chromosomes from multiple replications. So they prevent the chromosomes from losing base pairs that actually code for RNA and proteins. So telomeres protect the chromosomes from losing essential genes after each replication, because each replication, the DNA gets shorter since DNA polymerase can't finish the DNA replication at the 5' end.